welcome to the next chapter. It is Cell Physiology. Now, cell physiology is the study of the functions of the cell structures. Some of these functions carried out by these cell structures include the production of carbohydrates through the process of photosynthesis in chloroplasts, energy production by the process of respiration in mitochondria, and protein synthesis in ribosomes. For these physiological processes to occur, the cell must take in raw materials such as carbon dioxide, oxygen, mineral salts, and glucose. During the process, waste products are released. Some of these waste products are harmful and must be gotten rid of from the cell. Hence, there is a constant flow of substances in and out of the cell across the cell membrane in order to maintain life in a cell and an organism. In this chapter, we shall discuss the movement of materials in and out of cells. But first, let us look at the structure of the cell membrane and its properties. A membrane is a surface structure which encloses the cell and organelles. Such membranes include the cell membrane, tonoplast, nuclear membrane, mitochondrial membrane, chloroplast membrane, and others. These membranes have a common basic structure which regulates the flow of materials in and out of the cell or organelle. One such membrane which is important to a cell is the cell membrane, also called the plasma membrane. The structure of the cell membrane. The cell membrane, as seen earlier, is composed of a phospholipid layer sandwiched between two protein layers. Thus, the membrane is a lipoprotein layer. That means it is made up of a lipid and a protein layer. This gives the cell membrane a three-layer structure of about 7 nanometer thick, which is perforated by small pores that allow the passage of substances in and out of the cell, as appears on your screen. Properties of the cell membrane. The cell membrane bears some properties that allows it to perform its functions effectively. Semi-permeability. The pores that occur on a cell membrane allows molecules of small size to pass, but not those with large sizes. For example, when a cell is surrounded by a dilute sugar solution, water molecules will enter the cell but not the larger sugar molecules. The membrane is therefore said to be semi-permeable. However, the cell wall allows both water and sugar molecules to pass through because it has large spores, and it is referred to as being permeable. This property of semi-permeability allows the membrane to select what enters and what leaves the cell. Next, sensitivity to changes in temperature and pH. Cell membranes have a layer of protein, and these proteins are adversely affected by extreme changes in temperature and pH. Such changes alter the structure of the membrane, and therefore its normal functioning is hindered or affected overly. Possession of electric charge. A membrane has both positive and negative charges. These affect the manner in which substances are moved in and out of the cell. This property also helps the cell to detect changes in the environment. Physiological processes. Now, one major function of the cell membrane is its ability to control the movement of substances in and out of the cell. And this function is achieved through physiological processes that include diffusion, osmosis, and active transport. Diffusion. Particles in a gas or a liquid are constantly moving freely, bumping onto each other and bouncing off randomly. As this occurs, the molecules move from a region of high concentration to a region of low concentration. This results in the particles being evenly spread in the space available. Diffusion is the process by which particles move from a region of high concentration to a region of concentration. For example, the scent of a perfume sprayed in one corner of the building or a room will finally spread and be detected in every corner of the room. The perfume has spread via diffusion. When a piece of a potassium permanganate particle is dropped in a glass jar of water, you notice that after some time the purple color of potassium permanganate is spread throughout the water and eventually all the water turns purple. The explanation for this observation is that in the crystal, the particles of potassium permanganate are concentrated. The potassium permanganate crystal breaks away from the crystal, dissolving in the water and then diffuse through the water until they are evenly distributed. The difference in concentration of particles between the region of high concentration and that of low concentration is known as diffusion gradient. The steeper the gradient, the faster the diffusion. The role of diffusion in living organisms. Absorption of mineral salts. In plants, many salts dissolve in soil water to form ions, which are small enough in size to pass through the pores of the cell membrane of the root hair. The process will take place when the concentration of ions is higher in the soil water than in the root hair cells. Digested food, for example amino acids and glucose, diffuse across the walls of the ileum to the blood for transport to other parts of the body. Gaseous exchange in animals and plants. Plants take in carbon dioxide and give out oxygen in the presence of sunlight during photosynthesis. Some of the oxygen is used up in respiration and the excess is given out. At night, only respiration takes place, and therefore the plants take in oxygen and give out carbon dioxide as a waste product. These gases pass in and out of plants through stomata in the leaves and lenticels in stems by diffusion. In animals, on the other hand, several structures are used as surfaces for gaseous exchange through diffusion. These include the gills, the skin, the tracheal system, and the lungs. The structures provide large surfaces for gaseous exchange. In animals where blood is used for transport, respiratory gases, that is oxygen and carbon dioxide, move in and out of the body tissues by diffusion. Excretion of nitrogenous waste is another process that occurs through diffusion. And also, diffusion is one of the mechanisms involved in the transport of manufactured food from leaves to other parts of the plants. Several environmental factors affect the rate of diffusion. Now, factors affecting rate of diffusion, and the first factor is diffusion gradient. A greater diffusion gradient between 2 points increases the rate of diffusion. Increasing concentration of diffusing molecules also increases diffusion gradient with corresponding regions, hence increases the rate of diffusion. Surface area to volume ratio. The rate of diffusion in and out of the body depends on the surface area to volume ratio. The higher the ratio, the greater the rate of diffusion, and the lower the ratio, the less diffusion takes place. This implies that small organisms expose a large surface area to the surrounding when compared to big organisms. Therefore, small organisms depend on diffusion as a means of transport of food, respiratory gases, and waste products. Large organisms, in addition to diffusion, have transport systems to increase the efficiency of diffusion. These large organisms requires an elaborate transport system to bring the molecules of oxygen and carbon dioxide close enough to the cell surface for diffusion to take place, and that is the role of the circulatory system. Thickness of membranes and tissues. The thicker the membrane or tissue, the lower the rate of diffusion, and this is because of the distance to be covered by the diffusing molecules. However, if the membrane is thinner, the rate of diffusion is higher. Size of the diffusing molecules. Small and light molecules will diffuse faster than large and heavy molecules. Temperature. An increase in temperature increases the energy content in the molecules, and this causes them to move faster. Hence, an increase in temperature increases the rate of diffusion, while a decrease in temperature decreases the rate of diffusion. The next physiological process is osmosis, and osmosis is a special type of diffusion which involves a semi-permeable membrane. Now, if you consider a situation where we have two compartments divided by a semi-permeable membrane, and that one compartment is more concentrated than the other, the solid molecules are too big to cross the membrane. So what happens is that water molecules move from the compartment with low concentration of the solute to that with high concentration, and this is called osmosis. Thus, osmosis is the movement of solvent molecules like water from a region of low concentration to a region of high concentration across a semi-permeable membrane. Osmosis is a special type of diffusion. You can think of these compartments in another way. You can say that there is high concentration of water in the right compartment than in the left one. And so water diffuses to the left, only that in this case it is across a semi-permeable membrane. This movement will occur until the two compartments have the same concentration, and they are said to be isotonic. A low concentrated solution is described as hypotonic because it has less of the solid molecules but more of the solvent molecules. On the other hand, a highly concentrated solution is described as hypertonic. Why? Because it has more solid molecules but less solvent molecules. On the other hand, a pressure can be applied against the semi-permeable membrane to stop water molecules from the distilled water entering the concentrated solution or to stop osmosis from taking place. This pressure is referred to as osmotic pressure. The osmotic pressure of a solution is measured by an instrument called osmometer. Osmotic potential. Osmotic potential is the hidden pressure of a concentrated solution that only becomes real when the solution is separated from distilled water by a semi-permeable membrane. It is a measure of the pressure a solution would develop to withdraw water molecules from pure water when separated by a semi-permeable membrane. Water relations in animals. Now, the cell membrane of animal cells is semi-permeable, just like the whisking tubing, dialysis tubing, and cellophane. The cytoplasm contains dissolved salts and sugars in solution form. If an animal cell, for example the red blood cell, is placed in distilled water or hypotonic solution, there would be a net flow of water into the cell by osmosis. The cell will swell up and eventually burst. The cell membrane is weak and is not as strong and as firm as the additional cellulose cell wall found in plant cell. The cellulose cell wall in plants prevents the outward pressure and eventually prevents the cell from bursting. The bursting of the red blood cells is known as hemolysis. If a similar red blood cell is placed in hypertonic solution, then water would be drawn out of the cell by osmosis. The cell would shrink by a process called crenation. The behavior of red blood cells placed in various concentrations of solutions indicate that body fluids and the blood plasma surrounding cells must be kept at the same concentration, that is isotonic, as that which is found inside the cell. If this is not done, the cells will either gain water and burst or lose water and shrink, and their functions will be impaired. The body has a mechanism of keeping these concentrations constant within a narrow range. Water relations in plants. A plant cell has both cellulose cell wall and the cell membrane. The entire cell contains a vacuole with sap. The sap is a solution of salts and sugar and is bound by a membrane called the tonoplast. The cell membrane and tonoplast are semi-permeable, while the cellulose cell wall is fully permeable and thus allows solids and water to pass through freely. If a plant cell is placed in water or hypotonic solution, the water will move into the cell by osmosis and cause the cell to distend. The cellulose cell wall is rigid and does not allow the cell to burst as in the case of animal cell. As the cell gains more water, its vacuole enlarges and exerts an outward pressure on the cell wall called the turgor pressure. This pressure increases as more water is taken into the vacuole, which pushes the cytoplasm against the cell wall until the cell wall cannot stretch anymore. The cell becomes firm and rigid and is said to be turgid. When the cell wall is being stretched towards the outside, it develops a resistant pressure to the stretching that is equal and opposite to turgor pressure. This is called wall pressure. When a plant cell is placed in hypertonic solution, water molecules move out of the cell into the solution by osmosis. As the water moves out of the cell starts to shrink, becomes less rigid and flabby, and is said to be flaccid. If the cell continues to lose more water, its contents reduce in size and the plasma membrane pulls away from the cell wall towards the center. The process by which plant cells lose water, shrink and become flaccid is called plasmolysis. Plasmolysis can be reversed when a flaccid cell is placed in distilled water, and this process is called deplasmolysis. But still, osmosis has roles to play in living organisms. One is absorption of water from the soil. Root hair cells of a plant absorb water from the soil by osmosis, and this same process also helps in water distribution from cell to cell in the body. Next is support. Water taken into cells increases cell turgor, hence the cells become firm and rigid and therefore gain support. This type of support is important in seedlings, leaves and herbaceous plants which are less woody. Opening and closing of stomata. Gut cells synthesize glucose through the process of photosynthesis in the presence of light. As glucose accumulates in the gut cells, their osmotic pressure increases, and this enables them to draw water from adjacent cells by osmosis. This action results in the gut cells becoming turgid, causing the opening of stomata. At night, the gut cells do not photosynthesize, and therefore glucose levels goes down, leading to lowering of osmotic pressure. The gut cells lose turgidity, and therefore the stomata closes. Feeding in insectivorous plants. Now, insectivorous plants are plants that trap insects using special structures that suddenly change their target pressure when disturbed. The change in target pressure enables the special structures such as leaves to close, trapping the insect, which are digested to provide amino acids. Finally, osmoregulation. In kidney tubules of animals, water is withdrawn from the tubules through the tubular walls through osmosis. The water finds its way into the surrounding blood capillaries, and this helps the animal to regulate its osmotic pressure. Like diffusion osmosis is affected by several factors. Which ones are these? Concentration of solutions and concentration gradient. Now, osmosis is greater in solutions that have greater differences in osmotic pressure. The greater the concentration gradient between 2 points, the greater the speed of osmosis. Certain factors that increase the rate of diffusion also increase the rate of osmosis, provided that they do not destroy the semi-permeable membrane, and such include temperature. And the last physiological process is active transport. Now, diffusion and osmosis do not account for the transport of all substances across cell membranes. Substances such as amino acids, sugar, and many ions do not easily diffuse across the cell membranes, even if their concentration on one side is greater than on the other side. Other substances, although able to diffuse across cell membranes, do not meet the requirements of the cells. Active transport is a process that moves these substances across the cell membrane and against the concentration gradient, and this process requires energy to do so. It is suggested that there are carriers on the membranes which combine with substances being transported and then move them from one side of the membrane to the other. It is by active transport that seaweeds take up large quantities of iodine from their surrounding seawater and accumulate it in their tissues at a higher concentration than in the seawater. It is important to note that cellular intake of solutes is largely dependent on active transport. Roles of active transport: reabsorption of sugars and some salts by kidney. Useful substances are reabsorbed back into the bloodstream from the kidney tubules by active transport. Active transport is also involved in the absorption of some mineral salts from the soil by roots. It also plays a role in absorption of digested food from the alimentary canal of animals into the bloodstream. Active transport also plays a role in accumulation of substances into the body to offset osmotic imbalance in arid and saline environments. Finally, active transport is involved in the excretion of waste products from body cells and tissues.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 8. Anesthetics. One of the first anesthetics to be used during surgery was ether. In 1846, American dentist William T.G. Morton demonstrated that the pain and shock of medical operations and procedures could be lessened or even prevented with the inhalation of this organic compound. This made room for other drugs and anesthetics to be developed and facilitated the jobs of surgeons everywhere.